Hello everyone, my name is Zhao, and now I'm a research fellow in Professionals Group in NTU. Today I'm very honored to be able to participate in the AKC International Conference again, and I'm very happy to share with you the latest progress of our research group in pollen materials. Okay, let's go to the topic. The topic, the title of my topic is Pollen-derived hygroresponsive actuator. In nature, many organisms dynamically interact with the surrounding environment for specific function. Inspiration from such naturally biological systems has led to the development of a wide range of artificial soft actuators that can reversibly change their morphological or mechanical property in response to stimuli. Owing to their dynamic responsiveness, they have a wide range of potential applications that include soft robots, artificial muscles, sensors, and electric generators. Significant progress has been made in the development of actuator materials. However, the key components are derived from graphene-based material or synthetic polymers. Given the common limitations associated with these materials in terms of their high cost, strict reaction control, and the issue with the environmental sustainability, the quest for more cost-effective material from natural and renewable sources has accelerated. Although natural materials such as agarose and cellulose are used in a number of applications, they must be extracted and refined from raw resources at a considerable energy cost, and when actuators are constructed from such biopolymers, they typically exhibit weak actuation response, and their dynamic response is not conducive to fabrication of components that demand cycle variation in mechanical characteristics. In view of this consideration, it's highly desirable to explore the possibility of development and the widespread deployment of naturally abundant materials for applications requiring actuation and locomotion by resource to eco-friendly, sustainable, and cost-effective processing and fabrication methods. Here we describe a unique paper-like actuator made of plant pollen. Pollen grains are natural microparticles designed to encapsulate and transport plant genetic material and has been extensively studied on account of their steep property. In particular, pollen grains are composed of two mechanically distinct layers, a tough sperm pollen axis and a soft cellular intine. The double linkage of ester and acetyl groups contribute to the superior mechanical stiffness and the chemical inertness of the sperm pollen axis. Because of their toughness, pollens are also explored for applications such as drug delivery, microcapsulate, reinforcing fillers, and macromotors. Importantly, pollen-derived macrocapsules has been developed as non-allergic carriers in order to use advantageous properties such as high mechanical strength, chemical stability, and UV protection. Recently, we demonstrated that it is possible to convert hard pollen grains into soft, stimuli-responsive macrogel particles. However, the potential to develop other material forms based on natural and inexpensive pollen grain, thereby using their unique functional capabilities and applications, has not been investigated to date. In this study, we show a method capable of transforming soft pollen macrogels into a flexible and translucent two-dimensional paper-like material. The simple drying process is attributed to the activity of functional group during hydrogen bond formation, whereby pollen particles are assembled into macro-scale building blocks. Surprisingly, the resulting pollen papers display sensitive and reversible mechanical shape change in response to water vapor stimulation. First of all, we transform ultra-tough defected pollen grains into soft hollow material. Figure 8 shows sunflower pollen grains with spikes on their surface. This grain preserves their original 3D shape in dry and vacuum conditions as seen from SEM images. In this work, we soften the hard pollen grains by making use the traditional two-step soap making process. This process involves pollen shell extraction and the following incubation in an alkaline medium, KOH. The sample was incubated in fresh KOH for 3, 6, or 12 hours to facilitate extensive hydrolysis, then neutralization and centrifuge produce stable pollen macrogels, as shown in Figure C. In contrast to the original defected pollen particles, the incubated pollen particles swell in diameter. These particles experience significant structural collapse and flattening after natural drying, because of the enhanced exposure of their functional group during hydrolysis and owing to their softness. Pollen macrogel are like cellular or lignin and can serve as potential building blocks for the fabrication of higher order structural assemblies. Then we utilize a convention film-making strategy to assemble the pollen macrogels. Dilute pollen macrogels were easily cast into the mold. The construction of the pollen particle gradually increased with water evaporation, resulting in the accumulation of the particle and thereby leading to the continuous paper-like assemblies. The pollen paper exhibiting excellent mechanical flexibility and two different surfaces, a frosted top surface that was exposed to the air and a mirror-like bottom surface resulting from the contact with the bottom of the mold during water evaporation. FTR was used to determine the molecular structural shifts, which indicates improved exposure of functional groups active in formation of the hydrogen bond between the pollen particle surface. Consequently, integrated and freestanding pollen paper could be obtained. We can see the high transparency of the pollen paper. Moreover, the thickness of the pollen paper can be readily controlled by adjusting the weight of the original defected pollen grains, and an increase in KOH incubation time leads to a decrease in thickness because of prolonged alkaline hydrolysis of pollen particles. Differences in the appearance of the surface of the pollen paper can be tracked to their underlying microstructure. as shown in the SEM and AFM images. The top surface of the pollen paper had a non-uniform morphology and apparent structure with feature from the spikes, whereas the bottom surface was relatively much smoother. This finding was supported by analysis of the surface roughness using AFM, which revealed the significant difference in roughness between the top and the bottom surface. Additionally, with the increase in incubation time, the roughness of the top surface decreased, whereas the bottom surface of all samples shows near identical value of the low surface roughness. This suggests that increases softening of the pollen material particle with longer incubation and further indicates a decrease in surface roughness. The laminated nature of the pollen paper is obvious from the cross-section SEM image. Due to the exposure of hydrophilic functional groups during alkali hydrolysis, the pollen paper can absorb water. As a result, the surface area of the soaked paper was dramatically enlarged by more than 80% in the 6-hour sample. We found that a longer culture incubation time results in an increase in both surface area and weight, giving its lamellar structure and excellent swelling rate resulting from water absorption. The pollen paper appeared to exhibit a degree of mechanical responsiveness to humidity somewhat similar to that of many synthetic materials studied previously. Asymmetrical exposure to water vapor induced stronger interaction of hydrogen bonds between the water molecule and the surface of pollen particles. The following weakening of the original hydrogen bonds between the pollen particles leads to an expansion in the spacing between the pollen particles. Close packed and layered pollen particles, tens of micrometers in thickness, act as barriers that hinder and delay the diffusion of the water molecules. This unique structure, arising from the differential swelling of the pollen paper, induces internal stress through the thickness of the paper, which forces it to bend. As the water vapor is removed, the hydrogen bond between the pollen particle strengthens, forcing the paper to return to its original state. Continuous absorption of water leads to the pollen paper ultimately reaching a balance point. Any variation in humidity can easily shift the balance of saturation point and thereby reversibly change the weight of weight. In addition to the weight change, variation in humidity also had a strong effect on the mechanical property of the pollen paper. This suggests that moisture weakens the connection between the pollen particles and that the mechanical properties of the pollen paper are also strongly influenced by humidity. A strong stripe of pollen paper was subject to a fixed tensile strain of 1%. When humidity would decrease from 60% to 20%, contractile stress was generated within the sample. In turn, the imposed tensile stress had to be raised to 6.6 MPa in order to maintain the tensile strain of 1% when placed on a flat nylon mesh above a water bath. The pollen paper was observed to undergo a simultaneous and continuous flipping locomotion cycle as shown in the video. When exposed to a gradient in humidity level, the surface facing down expanded more than the opposite surface. This differential expansion caused the paper to curve up. Finally, the pollen paper unrolled upside down and then curved out from another perpendicular side to start a new cycle because of its reversibility. And a longer KH incubation time exhibited a more increase in the flipping locomotion speed. Next, we demonstrate mechanical actuation in pollen paper through control of such factors as KH incubation time and paper thickness. For this purpose, we design a simple device compressing a metal mesh and the beaker containing water that can create a humidity difference of around 20% between the metal surface and the ambient environment. When positioned in place with the top surface facing the water, all the sample exhibit a steady state curve within 80 seconds. Interestingly, when the bottom surface facing the water, we observe that all three samples displayed faster bending and unbending speed. We explained this differential behavior by noting that the smoother and more compact bottom layers of the pollen paper demonstrates a greater propensity to hinder the diffusion of water molecules as compared to the top surface. As a result, compared with the top surface, the bottom surface generally displayed increased curvature. Then we next demonstrate a potential application of pollen paper as a self-activating soft robot. For this purpose, we designed a biomimetic flower as shown in the video. After insertion into a well full of water, the biomimetic flower continued to bloom by the gradual absorption of water vapor that has been absorbed by the sponge from the well. Then we fabricate a simple binary pollen stripe that is capable of unidirectional working under cyclic variation in humidity. In this experiment, 3-hour and 12-hour pollen microgel samples were used to combine together. As shown in the video, when the biomaterial pollen stripe was exposed to cycle variation in humidity gradients, we observed the unidirectional movement of the biomaterial stripe. So, in summary, we create a economic wearable biomaterial-based soft actuator by a simple and eco-friendly process. And this actuator shows a strong mechanical actuation under changing conditions of humidity, and it is easily tolerable for specific application by varying the paper thickness and the KH incubation time. And it also has intrinsic differences in responsiveness between top and bottom surfaces. And we also create a walking robot. It is constructed by compressing two pollen sections of different KH incubation times. So at the end, we thank my supervisor, Professor Chum Nanjun, and the President, Professor Subras Raj, and their support and help in my work, and with all thanks to our all ETS group members. Thank you.